Welcome to Click for IB Biology. We're studying topic B3.2 transport, and in this particular video we're going to be looking at the movement of water through the stem of a plant in the process of transpiration. We have a PDF of this presentation linked into the description down below. Download that, annotate with your own notes, and then why not take the 10-question quiz with answers to be able to evaluate your own progress with this topic. So let's look at transpiration in the stem. And so the first thing that we need to do is to be able to look at the actual structure of the stem. And we begin by taking a transverse or cross section of the stem, as in AB. And here we've got two representations of that cross section, one with an actual light microscope specimen, and the one over here, which is a diagrammatic representation of those tissues. So if we actually start on the right hand side, notice that the stem in cross section, on the outside we have the epidermal cells, waterproofed to prevent water loss, and then the tissues which lie inside of that are known as the cortex, and those in the center of the stem are known as the pith. These are usually very large cells, and in fact we can see them here annotated as pith parenchyma cells. Arranged around the border between the pith and the cortex, there's a tissue, a stem cell tissue, which I'm tracing with my pen here. And you'll see that this is actually labeled as the cambium. Now, the cambium is responsible through its cell division for the production of the phloem tubes, which transport organic molecules, and the xylem, which transports water and minerals, and of course is the subject of this particular video. So the phloem tubes are on the outside, and the xylem tubes are towards the center and separated by the cambium. And so the cambium not only is responsible for the phloem and the xylem development, but of course as the lateral growth, as the stem grows, the cambium is constantly dividing to provide the cells for that as well. And we look over here at the light microscope representation of this, and we can see the arrangement of the xylem on the inside, the cambium in this location here, and the phloem, very small, are on the outside like here. There's the epidermal tissue on the outside like this. Now if we then look at this in longitudinal sections so that we can understand how water is moving through the xylem tissues, then with a powerful microscope, electron microscope, we would get a representation like we see on the right hand side of these longitudinal cells, and within those cells the contents of the cell have actually entirely broken down, so there's no cytoplasm or organelles remaining, only the cellulose cell wall. And in fact these cellulose cell walls are strengthened with rings of lignin, which are either going around in circles like so, and other times they actually will form spiraling structures like this. So there are a number of different patterns that we find within the extra thickening lignin rings. So that asks the question, why are these lignin rings necessary? And if we come across to this side of the diagram here, we remind ourselves that water is being removed by capillarity, hydrogen bonding, the cohesion between water molecules, as these water molecules are being evaporated from the leaf. And this evaporation pulls on the water molecules which are in the xylem, and those water molecules are bonded to each other by hydrogen bonds, a process, a structure known as cohesion. And so that as we actively draw the molecules from the top, this pulls on the column beneath and creates the so-called tension, which is known to exist within the xylem vessel. And if we think of our experience of drinking and sucking water through a paper straw, we know that if we suck upon the straw too strongly, the straw will actually collapse. And this is very much what's happening within the xylem, that if the water is being drawn too quickly, the xylem would collapse, but that's prevented by the lignin rings, which we have previously mentioned are strengthening the walls of this empty cell wall structure. Within those walls of the xylem, there are pits within that which allow water to be drawn out from this structure and into the surrounding tissues to maintain the turgidity of the plant tissue. And so the movement of water through the stem, through these vertical xylem structures, is characterized, as I said, as being under tension, under a negative suction pressure, which is generated by the loss of water in the leaf. We're going to go on in the next video to look at how that tension pulls upon the water which is produced in the root tissue. And that's the next video. And of course, chance to subscribe to the channel, that would be appreciated. And on the right hand side is access to the playlists so that you have quick access to the videos on the right hand side there.